Time. Right. Well, thank you, Larry, I think. At least he didn't cover the parts he said he was going to, so I'm pleased with that. But hopefully his introduction doesn't come out of my tongue. So I thought I would walk you through some of the exciting events in genomics. I was told this was a very broad audience, so some of you know some of these things, but I guarantee the stuff at the end you probably haven't heard of before unless you're on Twitter. So, one of the exciting things we're doing as an experiment, just at the other end of the mesa here, is building the first independent research institute on the UCSD campus. So this is right on the corner of the street at North Torrey Pines, and it's rising now out of the dirt next to the soccer field, and will be done about this time next year. And so I think it makes an interesting bookend with the Stem Cell Institute on the Mesa that hopefully will help drive some unique collaborations. This is also going to be as we know right now, the world's first carbon neutral building as a research building. It's not too hard to build a carbon neutral office building, but building a carbon neutral research building is much more complicated, than a lot of unique engineering went into this, including unique rooms for flow, fume hoods, water-cooled computers, etc. So it's going to be a living experiment as we go, but trying to show that buildings and labs and research can all live in a carbon neutral world, even though the Republicans on the planet don't think there's any need to. But does that help you know how I wrote it? So I'm going to talk about first digitizing biology, where we take what I view as an analog molecule, DNA with its 4 bases, and when we read that information and put it in the computer, we're digitizing it. So we're converting the ACs, Gs, and Ts into ones and zeros. And that's been going on now since 1977, when Sager's team sequenced the first DNA virus. The previous year was the first RNA virus. But it's obviously sped up tremendously as technology has changed, particularly over the last decade. Part of the challenge is understanding that digital information. And part of proving you can understand it is what the chemists have known for some time, and I learned here at UCSD in the chemistry department is you do proof by synthesis. So we tried to prove that, in fact, DNA, as we're reading the genetic code, in fact, contains all the necessary information. It's necessary and sufficient for life. So hopefully by the end of this lecture, you'll be convinced that DNA is the software of life, and that it contains everything needed for that. So let me walk you through that. As you heard, we sequenced the first genome of a living species in history in 1995, and 5 years later we're able to scale up for the first draft of the human genome. And 6 years after that, the first complete diploid genome. And to get a complete diploid genome, you have to have the complete set of chromosomes from both parents. And that's becoming a very important part of genetics. And most people who are not geneticists have known that their entire lives, because everybody wonders what traits you got from your parents, what traits you've given to your kids. But geneticists have never really looked at things that way, in fact, because they couldn't. And when they got sequenced, it was just looking at genetic changes without any assumption of where those changes came from. And so we're going on to look at ways to add complexity to our interpretation. Vanessa Hayes at the Venture Institute has been looking at global diversity, trying to trace back the origins of the first human genomes, doing the first African genome, the first Bushman genome, and then went on to do Desmond Tutu's genome, and comparing my genome to the first Han Chinese to the first Bushman. Everything came out in this sort of 1 to 3% scatter that we all expected. But when you look at just within Africa and look at the different populations, it's much more complex, in part because we all evolved from people who migrated out of Africa, but some of those people migrated back to Africa out of intermixing with developed populations. So the diversity within Africa is actually greater than the rest of the planet put together. And Vanessa's team has been tracing this back to a part in southern Africa with perhaps the most ancient human genome. So those are exciting developments, but understanding our inheritance comes from understanding your genetic code and your parents' genetic code. So we've managed to separate the genome into haploids. So in 2000, the big announcement on sequencing the human genome, in fact, we and the competing government labs only did half the genome. That's because it was expected to be about $1 a base pair, and